Hello, here I'm demonstrating the genSetView function in Human Epigenome Browser. The genSetView function allows you to focus on subset of genome. Either could be a collection of genomic coordinates or a meaningful collection of genes, or the other way as genes from a pathway. In order to use the genSetView function, scroll down to the control panel here. So by default, the genSetView interface is shown here. The genSetView can take two kinds of input. This is first the custom genset. You can enter your input in the text area here, one item each line. The input can either be gene name, as shown here, or genomic coordinates. So let's start by using this example input to run the genSetView. Just click the submit. So now the browser is running the genSetView. A new panel appears on top of the existing old submission interface. And let's take a look at the browser interface. Now the horizontal axis is being reorganized using the genes, and the genome heat map has been rearranged accordingly. So only the data over these genes are over each of those genes or genomic coordinates are being displayed, and same as any of the genomic feature tracks. So here you see this is a UCSC gene track, and only the genes overlapping the these intervals are being displayed here. And at this stage, you can still zoom in by dragging on this bar here and do the panning, so that you can zoom in to see detail of specific regions. Let's take a look at the new control functions after submitting the genSetView. These are basically the genset editing functions. So this category is the add, remove, where you can add new genes by entering them here, or you can mark the genes for removal. Remove genes from here. In order to remove genes, this is a list of currently displayed genes. In order to remove them, you have to click this button, and this marks this gene for removal. Once you have done your selection, just click the update. These two genes will be removed. Now these two genes are being removed from the view. The second is the change part, and this part actually function is available upon input. So let's just start over. If you want to turn off the genSetView, you can just click this button here. So I'm here start doing the genSetView again by showing the change part function here. So you see at the side of the input box, here is the change option. By default, it's showing the gene bodies, and if you click this, you will be able to choose different parts to be displayed for genes. No, currently this only applies for genes and doesn't apply for genomic coordinates. So if I select the 3KB promoter and gene body, here it shows it's gonna display the 3KB promoter and gene body here. Right, this is a change part function here. Let's use the other type of input. Here's the CAG pathway. This function will help you to query the CAG pathway, get one pathway, and do the genSetView on genes in that pathway. In order to find the pathway, you can enter the keyword here. You may enter only part of the keyword and click the pathway lookup. So this is all the pathways matching that keyword. Or if you know the pathway ID, you can directly enter the pathway ID here. And here, let's we don't have to change. You can still at this stage, you see you can still change the part of gene to be displayed here. The last function of the change part of genes is the custom region, which allows you to select an arbitrary range of flanking region for both upstream and downstream of certain type of genomic features. So you see right now it's selecting 2,500 base pair upstream and over 3,000 base pair downstream of certain. This is transcription start site, and you can define changes to the other two types of genomic characteristics. So let's change back to gene body. So let's click this pathway ID, which is glycolysis. The browser is retrieving genes from the glycolysis pathway, and will upon finish, we'll run the GenSat view on genes in this pathway. So it's finished, and you see there's a lot of genes here. And from this bar here, you will see each blue box corresponds to one gene in the current view. So this is a GenSat view. Yeah, the last function here is sorting, and you can rearrange the genes by various criteria. So the first sorting option is a chromosome order. Let's click update. So go to the genome heat map. You will see that now the genes on chromosome 1 are placed on the left, and of chromosome 2, then chromosome 3, 4, and this is the genomic coordinates very simply. And you can sort the genes by the length, long to short to long, or long to short. Here you see this is from short to long. On the left is very short, and on the right it becomes longer. So finally, you can do that. You can sort genes by average score of a certain track. So you need to select one track in order to do the sorting. You can either select one track from the panel here, or you can more conveniently you can go to the let's turn off this track. You can go to the genome heat map, select one, and we'll sort on it. So let's highlight this track here, change the color and also the height. So we will sort over this track. Just right click on it and select this option, select for sort or genpop. And now this name of the track will be displayed here. Just click the update. So now it's sorting the genes according to average value on this track, from low, which is on the left, to high, which is on the right. This is the sorting. So these are the GenSat view function, and you see the genplot. There's a genplot button, which is a very useful function coupled to the GenSat view. This function will be demonstrated in a separate section.